The history of neuroscience owes its narrative to individuals with great ideas, a few bitter controversies, and many animals. So I like to start this talk by asking you a question. To which of the following animals the progress of neuroscience owes the most? The pig, the frog, the chick, the dog, the rabbit, or the squid? Please choose one now, and we will see if your choice changes during the course of this talk. This talk is structured by highlighting 4 cardinal concepts that were pivotal in shattering previously widely held medical dictums, or that have shed light on apparently unsolvable problems. I will refer to these concepts as neuroscience landmarks, and they are brain, the seat of intelligence; action potential, the source of animal electricity; the neuron doctrine; and the last landmark is the ripples of the brain. It is important to stress that in choosing these landmarks, the characters I am about to introduce to you and the relevance I will give to each of these characters in this talk, I am subject to the bias brought about by my choice of literature, my limited historical education, conflicting information in the literature, and my geographical trajectory. Yet, I take solace on the fact that this bias has been afflicted others attempting to talk or write about the history of medicine. I will give you a well-known example of this bias. If you were to ask who discovered the blood circulation to a British physician, you will hear the name of William Harvey. The same question posed to a continental European physician will be answered with the name of Michael Servetus. The same question to an Arabic physician will be answered by naming Ibn al-Nafis. So, now that I have given you an example of the bias of others, I will start telling you mine regarding the history of neuroscience. So, let's go back to the landmarks I have chosen and elaborate on the first landmark, brain, the seat of intelligence. We all have sometimes been told, usually after making a bad decision, that we have made it thinking with our hearts. Why? Because of the ancient belief that there is a connection between the ticking of the heart, the ticking of the universal clock, and our actions. This belief is referred to as the cariocentric theory of thought. More recently, but still many centuries ago, the encephalocentric theory of thought came along, placing the brain as the thinking organ and as the master of our actions. It was Alcmaeon of Croton, the man most historians I have read credit with this shift. Alcmaeon of Croton was a pre-Socratic physician-philosopher, born, as you guessed, in Croton, a city in the Italian Boot, which, at the time of his birth and during his lifetime, was a Greek colony. In addition to putting forward the encephalocentric theory of thought, Alcmaeon of Croton linked the brain to vision, smell, and hearing. The association of these organs with the brain led him to surmise that perception was different from understanding, that understanding was the purview of the brain and perception was a function of the organs. In addition, he intuitively concluded that there had to be a passage connecting the organs of sight, hearing, and smell to the brain. The term Alcmaeon of Croton used to refer to this passage was poroi. Poroi, like most ancient words, has many meanings. The most common are pores, channels, path, openings, fibers, and threads. The last two, fibers and threads, apply mostly to biology. To appreciate the importance of Alcmaeon of Croton realization, we need to see them through the lens of history. And we will do so by going back to ancient Egypt. Judging by the frequency the Egyptians' hieroglyphic for brain is found encrypted in their papyrus, the Egyptians knew of the existence of the brain from very early on in their history. It is even possible, as some Egyptologists suggest, that the Egyptians had taken inspiration from the central region of a slightly cut brain, which I have enlarged here to carve the Eye of Horus. The Eye of Horus is a symbol venerated by ancient Egyptians of well-being, healing, and protection. As you can see, looking at the symbol of Horus side by side to the brain specimen, they have much in common. Here, I am indicating to the pituitary stalk that resembles the descending staff of the eyes of Horus. The brainstem has some similarities with this slanted structure. The corpus callosum, the structure connecting large part of the cerebral hemisphere with each other, resembles the upper curved line in the eye of Horus. The fornix and the anterior commissure, structures that connect both hippocampi and parts of the right and left temporal lobes respectively, resembles the upper eyelid and the tear duct. The thalamic mid-region resembles the eyeball, yet despite knowing about the existence of the brain, and even if using the central sagittal region of the brain as inspiration for the Eye of Horus, for the ancient Egyptian, it was not the brain but the heart that controls sensation, intention, and thoughts, thus responsible for the action of human on earth. Proof of this is found in this engraving from the Book of the Dead papyrus, believed to have been drawn over 1,000 years before Christ. In this engraving, we can see the portrait of Anubis, the jackal-headed deity assigned to accompany the dead in their passage towards the afterlife, leading the deceased to have the heart weighed against a feather. The feather represents Mat, the goddess of truth, justice, balance, and order, that you can see here represented with feathers adorning her arms. A second figure of Anubis checks the balance to decide the fate of the deceased, while the Ammit, portrayed with the head of a crocodile, 4 quarters of a lion, and hind quarters of a hippopotamus, also known as the devourer of the dead, stands ready to eat the deceased if found not worthy to see Osiris, who would otherwise welcome the dead in the afterlife. The last figure in the papyrus is Thoth, depicted as a man with the head of an ibis. Thop, the patron of scribes, is recording the result for posterity. Notice that what is recording is not the weight of the brain, but a jar, here enlarged, called the leb, which for the Egyptians represented the heart. Because for the ancient Egyptian, as previously mentioned, it was the heart, the organ responsible for the actions taken by humans while on earth. This cariocentric theory that started at the time of the Old Kingdom and probably before, persisted even through the time of Homer, who in the Iliad and the Odyssey, written around 700 years before Christ, does not mention the brain but the heart as the compass of human behavior. So it was not until Alcmaeon of Croton, as we previously mentioned, who was born in about 540 before Christ, wrote his famous treatise titled On Nature at apparently the ripe old age of 80, that the encephalocentric theory began to spread in the Greek world. Yet Alcmaeon's theory was not widely accepted by all Greek physicians, philosophers of the time, and it had to wait until the discovery of the structures connecting the brain and the limbs by Praxagoras, one of Alcmaeon's followers, for the encephalocentric theory to gain wide acceptance. Praxagoras described the structures communicating the brain with the rest of the body as minuscule arteries that had lost their lumen and become responsible for transmitting signals. Praxagoras went on to label these lumen-less structures with the Greek word of neuron. So what we know now as nerves were no longer called by the Greek word poroi, that, as you know, has many meanings, but by another Greek word, neuron, which only has two meanings, cord and sinew. As we will see later in this talk, Praxagoras' two brilliant insights: first, that connections between the brain and the body were made of solid cords, and the second, that the function of these solid cords was to transmit information, were for a long time forgotten. The next character I will introduce is Herophilus, who lived from 335 to 280 before Christ, and is considered the father of anatomy. The reason I drew a line after his name is because, as you know, historians often refer to ancient Greek personalities by adding to their name the name of the city of their birth or the name of the city where the personality worked and spent a significant part of their life. Following the first tradition, Herophilus is often called Herophilus of Chalcedon, the place of his birth, now a district in Istanbul. Based on the second tradition, adding the name of the cities where they had lived, Herophilus has been called Herophilus of Kos and Herophilus of Alexandria. But for this talk, we will refer to him as plain Herophilus. As you can see, now I am adding a question mark under Herophilus. The reason I place a question mark and not an image of Herophilus below his name at this point is because of the many different looking images identified with Herophilus in the literature. I am especially fond of the portrait used in Neuroglia, published in 2018. In it, he is called Herophilus of Chalcedon, but his image is the same as the one used for Galen, as you can see here labeled. So either Herophilus and Galen were fraternal twins born over 100 years apart, or this is an error. The same portrait of Herophilus used in neuroglia was used in the Journal of Neurology and Neuroscience in 2016. Here referring to him as Herophilus of Kos with a K instead of a C. A totally different image is presented in a paper published in Medicine 2015 titled Herophilus, the Great Anatomist of Antiquity. This same image is used by Wikipedia in a piece about Asclepius and looks a little bit as the image I use in this talk for Praxagora. The image published in a flyer in remembrance of Herophilus Day, just introduced in this frame, was used in the Encyclopedia Britannica for Aristoteles. Medscape used an image from Neurosurgical Focus published in 2004, which is unlike any other. The last image of Herophilus that I will show you is the one painted by Joseph F. Dove. This image currently housed at the Houston Academy of Medicine Texas Medical Center Library was reproduced in the Journal of Neurosurgery in 2005 and a sketch in a publication in the journal Anatomy and Cell Biology 2010. So, which of these images should take the place of the question mark in our talk? Well, I will make an executive decision and exchange the question mark for Joseph F. Dove portrait of Herophilus. Now that we have settled on a name and an image for Herophilus, let's just say a few words about Herophilus' contribution to neuroscience. Herophilus' contributions to neuroscience are many. Herophilus distinguished arteries from nerves, not an easy task in cadaveric specimen as you can see in this photograph. He distinguished sensory nerves from motor nerves and also found that damage to a motor nerve produces weakness. And in the central nervous system, he distinguished the cerebrum from the cerebellum and assigned different and specific functions to each of these structures. Herophilus assigned functions to the cerebrum were consistent with Maéon encephalocentric theory of thought. The final character in this series is Galen. Galen was born on the year 129 after Christ. Galen learned anatomy while taking care of gladiators. In this frame, you can see the portrait of Galen cutting the recurrent laryngeal nerve of a pig, which rendered the animal voiceless. Galen was, as Herophilus, a firm believer in the encephalocentric theory of thought. Galen's prestige and anatomical experiments buried the cardiocentric theory of thought for good and forever. So we have looked at the first neuroscience landmark, brain, the seat of intelligence, but before going on to the next landmark, I want you to consider the animal question I posed at the beginning of this talk. To which of this animal the progress of neuroscience owes the most? Well, I think at this stage we have to say the pig. But let's go on. Now we're going to look at the second landmark. Action potential, the source of animal electricity. Understanding this landmark is challenging. The term action potential and animal electricity may not be as familiar to you as are all other terms I have used in the other three neuroscience landmarks. Hence, as we talk about this landmark, I will take a few minutes to define these terms and also to explain some physiological and physics facts needed to understand this challenging landmark. If we are asked to name in 10 seconds as many sources of electricity as we can remember, most of us will probably name wind farms, water plants, nuclear stations, solar panel farms, batteries, and perhaps many others. But it is unlikely that we would name the action potential. Many years and many experiments were necessary to come to the conclusion that the action potential is the source of animal electricity. All in all, it took from 1664, when the issue of nerve conduction was addressed in modern times, till 1939, that is 275 years, the effort of many neuroscience and the sacrifice of many animals to have a complete understanding of the action potential and its role in animal electricity. The first neuroscientist I will introduce in this 275-year journey is René Descartes. Descartes, in about the year 1664, put forward the theory of vital force. The vital force was conceived by Descartes as a force not related to physics or chemistry, gifted to animals, including human, by God. Descartes considered the vital force to be the CSF ingredient that moved muscle and made the brain perceive sensations, feel emotions, and in human forge thoughts and govern behavior. He thought of the brain as a sophisticated pump, convening hydraulic pressure to the cerebrospinal fluid for the transportation of the vital force. He thought that the cerebral fluid traveled by invisible hollow conduits from the brain's ventricle to the muscle and from the skin and other sensory organs to the cerebral ventricles. In a way, Descartes' conclusions were that the nervous system and the circulatory system used the same mechanical principles. The brain was a pump of the nervous system, just like the heart was a pump for the circulatory system, and that the nerves, the conduits for CSF to flow, just like arteries and veins were the conduits for blood to flow. After Descartes, we will talk about Isaac Newton. Newton, in about 1680, argued that ether permeated the nerves and that the ether transmitted information by vibrations. Newton based his conclusion about the vibrating ether on the writing of one philosopher and the work of a physician. The philosopher was Pierre Gassendi. Gassendi's contribution to Newton's theory was to affirm that muscle was the source of force and that force was not brought to the muscle by the nerves. The physician contributing to Newton's vibration ether theory was Thomas Willis. Willis, in his book Anatomy of the Brain and the Description and Use of the Nerves, in addition to wonderful drawings of the inferior surface of the brain, depicting in one of these drawings what later came to be called the Circle of Willis, he published exquisite topographic drawings of most major nerves, as the one that you see here of the glossopharyngeal, vagus, and accessory nerves. The weight of Newton's reputation in the scientific community was such that the vital force and hydraulic theory sponsored by Descartes after Newton's assertions were discarded. After Newton, we will talk about Luigi Galvani. About 40 years after Benjamin Franklin had proven the presence of natural electricity, Galvani discovered animal electricity. Luigi Galvani, as you can see in this portrait, experimented with frogs. In one of his initial experiments, he dissected a frog and discarded the head, the four legs, the trunk, and most of the spinal cord, thus creating a preparation that consisted of the lower section of the spinal cord, the sciatic nerves, and both hind skinless legs. He then touched the sciatic nerves with a copper wire and one foreleg with a thin wire, initially keeping both wires apart. In this situation, nothing happened. But when he brought them together, the legs contracted. Galvani interpreted the contraction of the frog hind legs as a result of an intrinsic electricity present in the muscle that was released by the interaction with the metal. Hence, he used the term animal electricity to refer to it. This discovery and the reasoning Galvani attached to it led to a big controversy. The controversy was between Galvani and Alessandro Volta, a famous physicist. Galvani arguing that the reason the frog leg jerked was because nerve and muscle generated their own electricity. Volta saying that the source of electricity was the metals, and both the nerve and the muscle just conducted and reacted to it. Volta based his assertion in two experiments. The first one was Benjamin Franklin's experiment. Volta argued that the string holding the kite did not produce electricity and still the key became hot and red. Volta considered that the string behaved like the nerve, conducting electricity, and the key as a muscle reacting to it. The second experiment was his own, the electric pile. The electric pile Volta had just created consisted of units made of discs of two different metals, copper and zinc, and a cloth soaked in salt water between them. These units were piled on top of each other with conductive wires attached at both ends. This device produced electricity without the need of animal tissue. The sound conclusions Volta arrived based on the two experiments we have just mentioned were counteracted by a new experiment made by Galvani. In this new experiment, the metal wires were both in contact with the sciatic nerve and the frog leg would still contract. But this new experiment did not convince Volta, but neither Volta's argument convinced Galvani. So, which one of the two was right? At the time, Volta was considered to be on the right side of the argument. Proof of this is that it was Volta and not Galvani, the scientist invited to display his experiment in front of Napoleon and perhaps even Benjamin Franklin. But Galvani, years later, in 1784, anonymously published a book titled on the use and activity of the conductive arc in the contraction of muscle. In this book, he described muscular contraction without the need for metallic contact. He did so by going back to his favorite frog leg preparation and inserting the sciatic nerve of a single frog leg preparation into one of the hind leg muscle of his original preparation, and noticing that the odd leg would jerk immediately when the sciatic nerve was plugged into the muscle. This should have been proof enough of the presence of animal electricity, but because the frog leg specimen was mounted using metals and that other scientists were unable to consistently reproduce Galvani's finding, it was not. After Galvani, I will mention Carlos Matteucci. Matteucci confirmed that animal electricity was present in muscle and that electricity could travel from one end of the muscle to the other. Matteucci made these discoveries based on three facts. First, electricity can be measured in muscle preparation. Second, muscle produces electricity if one electrode is touching the intact end of the muscle mass and the other one is touching the cut or injured end. And the third fact was that frog hind leg assembled serially, placing the intact part of one thigh in contact with the cut part of its neighbor, produce incremental amounts of electricity. I will be illustrating these three facts shortly, but before I do so, I need to tell you about the first instrument used to measure animal electricity. This instrument is the frog leg galvanoscope. Here you can see the drawing of a frog leg galvanoscope inside a tube. The reason for the tube is to keep the specimen bathed in normal saline to prolong the muscle tissue capacity to contract. The word galvanoscope has two parts, galvano and scope. The word galvano was used to name this instrument in honor of Louis Galvani, and scope because it was read by gross observation. The observation of the strength of the jerking of a relatively fresh dissected frog leg. In this frame, I am indicating to the frog leg muscle, here to the sciatic nerve, and here to a wire. The wire is wrapped around the sciatic nerve, as you can see. The frog leg galvanoscope was, as we previously mentioned, the first instrument used to measure animal electricity, that is, to measure voltage in animals. So now that we know what a galvanoscope is and what it is used for, we go back to Matteucci's experiment. The experiment consisted of placing 4 frog thighs in series, the intact surface of a frog leg thigh preparation contacting the cut or injured surface of the neighboring frog leg preparation, as you see in this illustration. Then touching the cut surface of the first muscle with the tip of a thin wire, as indicated by the arrow, and touching the other end of the preparation, the intact side of the last muscle, with one end of a copper wire, as indicated by the arrow. He then brought the wires together at one point and went on to insert the end of the wire of the galvanoscope into the different frog leg muscles, starting by the one closest to the thin wire. And when he did so, he noticed a minimal twitch. He did the same in the second thigh, and there was a bigger twitch. He did the same in the third thigh. The twitch was bigger, and did the same in the fourth thigh, and the twitch was even bigger. The successive increase in the twitches was the clue. Matteucci, based on these observations, concluded that since he had not changed the size of the wires, the successive increase in the force of the frog leg contractions must come from muscle-generated electricity. Thus, Matteucci confirmed the presence of galvanized animal electricity, and he called this animal electricity the injury current. He called it the injury current because it was only present when he placed one of the electrodes in the injured or cut surface of the muscle and the other one in the intact surface of the muscle. Matteucci then decided to confirm the presence of injury current in muscle and detect injury current in nerves by using a new, more sensitive instrument than the galvanoscope, an instrument able to react to lower voltage currents. This instrument was the Nobili galvanometer. The Nobili galvanometer was designed by Matteucci's teacher Leopoldo Nobili and was certainly a better tool to study the injury current than the galvanoscope. As I did when for the first time I mentioned the galvanoscope, I will take a few minutes to tell you about the principle behind the Nobili's galvanometer. I will do so by sending you back in time to your childhood, specifically to your first physics class. I will do so to remind you of 6 basic concepts about electricity and magnetism that are essential to understanding the workings of the Nobili galvanometer, as well as all other galvanometers. The first of these basic concepts is that a galvanometer is nothing more than a compass, adapted to indicate positive potentials and negative potentials, measured in volts. Hence, galvanometers these days are often referred to as voltmeters. The second concept is that if you bring a magnet close to a galvanometer, the magnetic force will move the magnetized arm of the needle towards the magnet. The third concept is that if we bring 2 magnets close to the galvanometer, the magnetic force will be stronger, and the magnetized arm of the needle will move even further towards the magnet than when it was only one magnet. The fourth concept is that if we start with the magnetized arm of the needle drawn towards a magnet and we invert the magnet, the force of the magnet will move the needle in the opposite direction, as you can see in this frame. The fifth concept is that if we connect a battery, let's say a 1-volt battery, to a wire wrapped around an iron rod, as shown here, the electricity traveling in the wire in one direction will produce a magnetic force just as the magnet did, and draw the magnetized tip of the needle towards it. But if we invert the battery, as you can see here, the new magnetic force generated will displace the needle in the opposite direction. The sixth concept is that a 1-volt battery connected to a wire wrapped 7 times around an iron rod produces a magnetic force capable of displacing the magnetized arm of the needle half the distance than a 1-volt battery connected to a wire 14 times wrapped around the same size iron rod, as indicated here. Now let's go back to Matteucci. We left him trying to use the Nobilis galvanometer to confirm the presence of muscle injury current and attempting to find the same current in nerves. The Nobili galvanometer consists, as you would expect, of an electrical wire wrapped around and around, as illustrated here, an iron rod, one end of which can be seen in this drawing, and a dial with a magnetized needle mounted above it, adapted to measure positive and negative potentials in volts. Thus, the Nobilis galvanometer measures electricity, that is voltage, by the displacement of the magnetized needle tip induced by the magnetism produced by electricity. So Matteucci recorded the injury current from a single frog leg specimen and logged, let's say, 1 millivolt. He then recorded the electricity from a 4 frog leg thigh pile and logged, let's say, 4 millivolts, thus confirming the presence of animal electricity and muscle injury current potentials. In this frame, I have added the first drawing of a frog pile from an early Matteucci publication. The name frog leg pile was used to describe this contraction because of the similarities with the electric pile designed by Volta. So, what is injury current? To explain it, I once again will use the time machine to send you back in time, but this time to a more recent past, to high school biology 101, to remind you of 2 basic facts necessary to understand the injury potential. The first one is that at rest, the extracellular medium just outside the cell membrane is positive charge when compared with the intracellular medium just inside, abutting the cell membrane. I will illustrate these statements in the next few minutes. In this frame, I am representing a cell, mainly a muscle cell or a nerve cell. The cream color area inside the white line corresponds to the intracellular medium. The white line represents the cellular membrane, and the blue area just introduced represents the extracellular medium. At rest, an explorative electrode connected to a voltmeter, coupled with a reference electrode both in the extracellular space, will register no electricity, that is, no voltage. A switch in electrode arrangement, as illustrated in this frame, that is changing the relation of the exploratory and reference electrodes, will also record no voltage. The same will occur if both electrodes are in the intracellular space, regardless of the relative positions of the exploratory and reference electrode. No voltage will be recorded. But if we place the electrodes separated by the cell membrane, one outside the exploratory electrode and one inside the cell, just abutting the cell membrane, the reference electrode, we will register voltage. This potential will displace the voltmeter needle towards the positive side. We call the force driving the needle, as you would expect from our prior explanation about galvanometer, a positive potential. This is so because the explorative electrode placed outside the cell membrane will record a more positive potential than the reference electrode, which is recording from the inside. But if we place the explorative electrode inside and the reference electrode outside, we will record a negative potential, because the potential inside the cell recorded by the explorative electrode is more negative than the potential outside the cell recorded by the reference electrode. So with this information in mind, we can visualize a cell at rest as having negative charges inside close to the membrane and positive charges outside close to the membrane. So how does this help us understand what is the injury current? To answer this question, I need to remind you of the second fact you needed to remember from your 101 biology class. This fact is that in a situation as illustrated in this frame, since we are recording with both electrodes outside a resting cell, we would register no voltage. If now we were to remove the wall of the cell where the reference electrode is located, as illustrated here, once the isolating wall is gone, the reference electrode will record a negative potential first, and shortly after, due to the balancing of positive and negative charges, a neutral potential. Either way, the exploratory electrode, now referred to a neutral reference and initially to a negative one, will record a positive potential. It is this positive potential that came to be called the injury current. So the injury potential is nothing more than a positive potential that arises from recording many cells with the exploratory electrode facing the intact cellular membrane side and the reference electrode facing the wall-less side of the cell. Going back to Matussi's experiment with the frog leg muscle mass, as you can see here illustrated, the exploratory electrode rests on the intact outer surface of the muscle, whereas the reference electrode rests on the cut surface of the muscle fiber. Once Matussi confirmed the presence of injury current in the muscle, he thought of doing the same in nerves. He used 4 frog sciatic nerve segments in series, as he had done before with the frog leg thighs, but the needle of the galvanometer did not move. He increased the number of sciatic nerve segments, but again the needle did not move. He was sure that nerve had injury current, but wondered why he was unable to find it. Frustrated, he decided to go into politics, and he became senator for life and minister of education in his native Italy. This brings us to Dubois-Reymond. In a book about his life, written by Gabriel Finkelstein, he labeled Dubois-Reymond as the most important forgotten intellectual of the 19th century. This neglect may have had something to do with his personality. He was accused at times of sounding conceited. He would tell his students in Berlin, only half jokingly, there are 2 outstanding physiologists in the world. The other one is in Leipzig, referring to himself and probably to Carl Friedrich Wilhelm Ludwig. Statements like this one did not make him popular among other physiologists. At the time, he was pugnacious, as portrayed in this newspaper clipping of the time, where he is portrayed as a cock arrogantly looking at Ernst Schwinninger. Dr. Schwinninger had been named chairman of dermatology at the University of Berlin in 1884 by Bismarck, a powerful German politician and a grateful patient. This appointment was against the wishes of the medical faculty and, according to Dubois, despite Schwenninger's lack of qualifications. Dr. Schwenninger was not the only physician he argued with. He had a bitter argument and rivalry with Matteucci. This rivalry was amply described by Finkelstein in his book and in this paper. Suffice to say that the exchange of words between them was not pretty, and at times Dubois' comments were right down insulting. A similar controversy arose between Dubois and Lubindar Hermann, one of his students. The final comment I will make about Dubois' personality is a quote from one of his contemporaries, who wrote that the only thing better developed in Dubois-Reymond than his capacity to make new friends was his ability to lose old ones. Well, regardless of his personality, his ability as a neuroscience were never questioned. His contribution to neuroscience was monumental. Dubois-Reymond was a brilliant experimentalist. He used frogs, as most physiologists did in those days, to conduct his experiments. I'd like to call your attention to this cover area of the frog, which is further illustrated here. Notice that the site of the experiment corresponds to the area of the sciatic nerve. The sciatic nerve, as we know, connects the spine with the leg muscles. Dubois-Reymond was looking at this area, hoping to answer the same puzzling question that baffled Matteucci. Is the injury current present in nerves? He was aware, because of Matteucci's findings, that muscle could conduct and generate electricity. Dubois was also aware, as was Matteucci, that electricity could be transformed into a magnetic force by wrapping a metal wire around an iron rod and allowing electricity to go through the wire. And he also knew that increasing the number of turns of the wire around the iron rod would result in an increased magnetic force. With this last fact in mind, he settled to create a better galvanometer. So here we have Nobilis galvanometer. What Dubois did was very logical. He created a new galvanometer with two major improvements. The most significant of the two was an increase in the number of turns of the wire around the iron rod, and the second improvement was that he created a dial with a needle requiring less magnetic force to be moved. Compare the space occupied by the coil wire element in the Nobilis galvanometer with the space occupied by the coil wire element in the Dubois galvanometer. As you can see, the size of the coil wire element in Dubois galvanometer is bigger. It is bigger because of the greatly increased number of wire turns around the iron rod. Now compare the size of the dial and the needle in Nobilis galvanometer with the one in Dubois galvanometer. Notice that the dial and the needle of the Dubois galvanometer, here schematically represented, are smaller than the dial and the needle in the Nobilis galvanometer, hence more likely to move by a weaker magnetic force. Then Dubois, using this improved galvanometer, conducted an experiment that influenced the course of neuroscience forever. The diagram represented here shows the main elements of the experiment. Here illustrated is a placeholder that, in addition to wires connected to an energy source like an electric voltaic pile, in other words a battery, these wires at this point are not touching, hence not forming a circuit, and therefore not injecting electricity into the system. These are the reference electrodes. These are the exploratory electrodes. Each pair of electrodes is connected to a galvanometer, here only represented by their dial, and the needle adjusted to the middle between the positive and negative signs. I have now added a green color line to represent a sciatic nerve segment. But keep in mind the arrow indicating in this dial to make sure you notice that as soon as the sciatic nerve segment was placed, the needle turned positive. Now let's look at what has just happened. The injured or cut side of the nerve are in direct contact with the reference electrode. In this frame, I have represented the cut side of the nerve as containing myelinated and non-myelinated axons, as well as epineuron. The epineuron is represented by the outside green color circle. The arrows are now pointing to the sites of contact between the explorative electrodes and the non-injured nerve surface, the intact epineuron. Now I want to call your attention again to the position of the needle in the dials. Notice that both dials have been showing a positive deviation since the segment of the sciatic nerve was introduced. When Dubois saw this sustained positive deviation, he realized that he had found the nerve injury current. But then he took a giant step forward. He went to the energy source element that so far had used only as a placeholder and closed the circuit, thus injecting an electrical current in the middle area of the nerve segment. And notice when he did this that the tip of the needle briefly deviated towards the negative side, quickly returning to its previous positive position. He reasoned that this brief negativity represented the passage of the electrical current through the nerve abutting the exploratory electrode, triggered by the closing of the circuit. Dubois called this phenomenon the negative Schwankung. And realizing the magnitude of this finding, he wrote in his diary, "If I do not greatly deceive myself, I have succeeded in realizing the 100 years dream of physicists and physiologists, to wit, the identity of the nervous principle with electricity." He used the term identity of the nervous principle to imply how nerve produce electricity. And indeed, he had found the neurophysiology Rosetta Stone that in the future would help neurophysiologists understand the connection between will and action and between perception and understanding. He had not only recorded the nerve injury potential, but the mechanism by which electricity is generated in animals. So, what is the negative Schwankung? As I did when I mentioned the galvanoscope, the galvanometer, and the injury current for the first time, I will take a few minutes to tell you about the basic principles necessary to understand the negative Schwankung. To do so, I will again use the time machine and send you back to Biology 101 to remind you about 4 basic principles. 2 of them I have already mentioned, related to neuronal behavior, necessary to understanding the negative Schwankung. The first principle we have explained before the last time I sent you through time to Biology 101, but I have displayed in this frame the final illustration to remind you of it. At rest, the extracellular medium just outside the cell membrane is positively charged when compared with the intracellular medium just inside the cell membrane. The second principle is a bit more complicated, so I will first explain it and then illustrate it. During activation, the extracellular medium just outside the nerve cell membrane is negatively charged when compared with the intracellular medium just inside the cellular membrane. In an activated neuron, recording with both electrodes in the extracellular space or in the intracellular space, as it was done in the resting cell example, will record no voltage. But recording with an explorative electrode just outside the cell membrane and the reference electrode just inside the cell will record a negative potential, reflecting the negativity of the extracellular medium just outside the cell membrane. But if the position of the electrode is changed so that the explorative electrode is in the inside abutting the membrane and the reference electrode just outside, a positive potential will be recorded. This phenomenon can be represented as illustrated in this frame. The extracellular medium just outside the cell membrane is negatively charged and the intracellular medium just inside the cell membrane is positively charged. The third principle we also explained previously, but it was explained related to the cell. This time we will extrapolate the explanation to the axon. At rest, the extracellular medium just outside a patch of axon membrane is positively charged when compared with the intracellular medium just inside the patch of axon membrane. The fourth principle follows the same trend. It was initially explained for the cell body, and here we will extrapolate it to the axon. During activation, the extracellular medium just outside a patch of axon membrane is negatively charged when compared with the intracellular medium just inside the patch of axon membrane. I will now bring these four principles together and illustrate them in the next few frames. Nerve cells have axons, the stuff nerves are made of. At rest, as we have just seen, nerve cells have positive charges outside the cell membrane and negative charges inside. Axons, as neurons, at rest are positively charged outside the membrane and negatively charged just inside the membrane. In this frame, I have represented an activated cell. Notice that the charges outside the cell body are negative and inside positive. The activation in axons occur in patches, physiologically always starting in the axon region nearest to the cell body. The changes in the adjacent patch of the axon close to the body of the neuron, as indicated here by the red arrow, change polarity. As we can see in this frame, notice that the previously positive external surface medium has become negative and the inner surface of the membrane positive. Notice that while this change in potential occurs in the axon, the neuron reverts back to its resting state, and that simultaneously with the neuron body reverting back to the resting state, the patch of axon ahead reverts potential, making the membrane patch just ahead negative in the outside and positive in the inside, while the patch of the membrane just behind reverts to its resting state, as the cell previously did. This process continues with the following patch of axon becoming activated, as illustrated here, moving distally along the axon. So now we can go back to the question, what is the negative Schwankung? As you recall, this frame illustrates the initial findings in Dubois' experiment. When no charge is being injected to the nerve segment, as illustrated by the open circuit, and the galvanometer dials indicating a positive charge corresponding to the excess of positive charges in the intact superficial segment of the nerve when compared with relatively excess of negative charges present in the intracellular compartment of the many axons exposed to the electrode by the cut surface of the nerve. This is the nerve injury potential. Now, when the circuit is closed and electricity is injected in the middle of the nerve segment, the surface negative wave indicated by the red thunders as it goes through the patch of nerve above the exploratory electrode, the needle in the dials shift negative. Then, when the surface negative waves are no longer traveling in the patches of the nerve abutting the exploratory electrode, the needles shift back to positive. It was this shift first from positive to negative, as indicated by the green arrow in the dials, and from negative back to positive, that Emile Dubois called the negative Schwankung. Negative because it trends from positive to negative, and Schwankung because in German it means variation, implying a sudden and brief change. Dubois, although not being able to provide a reason for the negative variation beyond the electrical explanation, suggested that the energy producing the negative variation was chemical in nature, and it turned out to be true. But it was not Dubois, but one of his pupils, Julius Bernstein, here shown as a relatively young man, and here, when much older, that took further steps elucidating the chemical nature of the electrical phenomena. As a young man, Bernstein worked in Dubois' lab and built an ingenious machine that he called the differential rheotome, meaning the cornet slicer, a device used to measure the duration of a negative variation. I will not attempt to describe it because I do not understand how it works. Using this machine, he recorded a tracing of the negative variation. Bernstein initially used Dubois' terminology, calling it the negative Schwankung, and it was probably him that first called this phenomenon the action potential. It was also Bernstein that postulated the role of potassium in the production of the resting and action potentials. Years later, Ernest Overton recognized that it was in most instances the interaction of sodium and potassium that produced the action potential. But we have to wait many years until 1939 for Alan Hodkin and Andrew Huxley to actually trace the first intracellularly recorded action potential, meaning being able to insert and place an exploratory electrode just inside the cell abutting the cell membrane without injuring the cell or the axon. This technique was possible thanks to the squid, especially to the giant axon of the squid. These neuroscientists introduced into the giant axon of the squid, which some are as thick as a human hair, a very thin needle. This achievement was possible thanks to two ingenious devices. One that allowed the simultaneous viewing of the needle penetrating the axon from two angles, the front and the side, thus avoiding perforating the axonal cell membrane. And the second device, a recording unit that allowed the actual tracing of the action potential to be recorded. Alan Hodkin and Andrew Huxley formed one of the most productive and influential collaborations in the history of neuroscience. The recording of the first intercellular action potential earned Alan Hodgkin and Andrew Huxley in 1963 the Nobel Prize in Physiology or Medicine, and also a knighthood. It is to their credit that, despite frequently being questioned as whose idea was to use the axon of the squid, they had never said. Now that we have finished talking about the action potential, I'd like to pose the animal question to you again. To which of these animals the progress of neuroscience owed the most? I am torn between the frog and the squid. The third landmark is the neuron doctrine. The neuron doctrine was the brainchild of Santiago Ramón y Cajal. But before talking about Cajal, the neuroscientist, I will tell you about Santiago Ramón, the man. Santiago Ramón y Cajal, Ramón being his father's first last name and Cajal his mother's first last name, studied at the University of Zaragoza. Upon graduation, he enlisted in the Spanish army as a medical doctor. He was sent to Cuba, at the time fighting for its independence from Spain. In Cuba, he did not get shot, but contracted malaria, and as a result was discharged from the army. Back in Spain, he developed a passion for the microscope, which went on throughout his whole life. But he also had other passions. He drew and he painted from very young. These drawings are from when he was 9 years of age. He took photos. This one he took with his entire family, and this one many years later with his daughter Paula. He practiced hypnosis. He used hypnosis to induce anesthesia during his wife's delivery of his sixth child. He also used hypnosis to successfully treat patients with a variety of maladies. After a while, his fame as a hypnotist and the fact that he never charged to do it brought him a large clientele. He considered the potential of developing a large clinical practice using this modality of treatment, but decided against it. At the end, as he wrote years later, he experienced with hypnosis left him with a feeling of surprise and disappointment. Surprised that it worked, and disappointed that it did, because as he wrote years later, it revealed to him the degree to which the much praised human brain, masterpiece of creation, suffered from the enormous defect of suggestibility. Santiago Ramón y Cajal wrote many books about science and children and adult literature. Among all of the books he wrote, one of them, the one which cover is in this frame, The World View at 80 years of age, I have read 3 times. The first time, it was during a winter break while as a young medical student in the University of Zaragoza. I did not find it very engaging. The second time I read it, I was 45 years of age. It was during a week while staying at a Miami Beach hotel with the occasion of a Nicklaus Children's Hospital, former Miami Children's Hospital, pediatric course being broadcasted to South America. I thought at the time that it was a good read. The third time I read it, a few months short of my 70 years of age, during a trip to Barcelona, I thought it was brilliant. I greatly recommend it as a must read if you can read Spanish and you have passed your 69th birthday. I will now talk to you about Cajal as a neuroscientist. The neuron doctrine states that the nervous system is made up of discrete individual cells. He made this remarkable observation by studying the cerebellum of chicks and embryos of different animals, using a recently developed histological stain technique. An epiphany, intuition, and chance led to this remarkable observation. The epiphany was his decision to look at immature instead of mature tissue. This idea came to him when looking at a forest with old and recently planted pines. He realized while gazing at the full-grown forest that he was unable to identify the branches of the pines beyond those at the edge of the forest. But when looking at a young forest in the nursery state, he was able to see the branches of all the pines. Hence, why not look at immature tissue when looking at the nerve cells, trying to see all its parts? The intuition was to look at the cerebellum. The choice of the cerebellum provided him with a tissue of central nervous system that undergoes significant histological changes late in embryological life and beyond. I will show you two examples of these changes. One of these changes is the distal migration of the climbing fibers. Here you can see the climbing fibers moving from the body of a Purkinje cell in a very premature tissue to the proximal dendrite region in a premature tissue, and finally just above the mid-Purkinje cell dendrite region shortly after term. The second example of late embryological development is still going on in early postnatal life. It is the internal migration of the external cerebellar granular layer. As you can see in this Cajal illustration, indicated by the numbers, the cerebellar neurons first, when immature, are present in the cerebellar external layer, whereas in the mature cerebellum they are present deep in the cerebellum. The third factor propelling Cajal to the neuron doctrine was chance. A chance trip to Madrid in 1887. During this trip, Cajal spoke with Dr. Luis Cimarro La Cabra. Dr. Cimarro told Cajal about a new staining technique called Reazione Nera discovered by Camillo Golgi. Hearing about this technique during this trip was the beginning of a lifelong affair with this stain, which Cajal greatly improved in subsequent years. The epiphany, intuition, and chance, followed by hard work, led to the publication in the newly minted self-financed Revista Trimestral de Histología Normal y Patológica in May of 1988 of an article about the structure of birds' nerve centers. After this publication, there were many others in the same and other journals with detailed illustration that show without any doubt that the nervous system is made up of discrete individual cells. This drawing represents a good example of it. The arrows indicate the body of several neurons, and now to the gaps between them. These gaps were called by Cajal nervous articulations. Neither the term neuron that was introduced by the German anatomist Heinrich Wilhelm Waldeyer in 1891, likely based on Praxagoras' use of this word, nor the term synapse, introduced years later by Charles Sherrington in Foster's textbook of physiology in 1897, existed at the time of Cajal's initial publications. But it was Cajal publications that nested all these terms and earned him the presentation by King Oscar II of Sweden of the Nobel Prize in Physiology or Medicine in 1906. In this frame, you can see the Nobel Prize diploma. Interestingly enough, the prize was shared with Camillo Golgi, despite Camillo Golgi's opposition to the neuron doctrine. Camillo Golgi was not alone in his opposition to the neuron doctrine. Neuroscientists at the time were divided in two camps. Reticularists. The reticularists believe that the brain nervous system consisted of processes of contiguous cells fused to create a massive meshed network. Neuronists, on the other hand, believe that the nervous system was made of discrete individual cells separated by gaps, as Santiago Ramón y Cajal did. The reticularists, among which we have Camillo Golgi, who, as we just mentioned, shared the Nobel Prize with Cajal, discovered the ratione nera in a manner not unlike Fleming discovered penicillin, by a mixture of chance and curiosity. To chance we owe that one day, as Golgi was working in his laboratory, and a piece of nervous tissue that had been hardened by exposure to potassium dichromate fell into an open jar of low concentration silver nitrate. To Golgi's curiosity we owe that he did not throw the piece away in the garbage, but instead he looked at it under the microscope. What he saw amazed him. Some nerve cells and their fibers were clearly visible. He went on to replicate the technique, and after many trials he published it, calling it, as previously mentioned, reazione nera. Based on this technique, Golgi made exquisite diagrams of the central nervous system, as the one you can see here. And here, I think these drawings are much prettier than many of Miro's paintings now displayed in museums all over the world. In addition to Camillo Golgi, in the reticularist camp, we have Otto F.K. Daters. He, using different staining methods, created detailed drawings of the nervous system, as you can see in this frame. He actually, without realizing it, drew the presynaptic junction regions that he described as triangular regions, but he did not interpret them as junctional components. Daters only lived to the age of 29 years. Looking at his life cut short by typhus, we cannot help but wondering if not had it been for his early and untimely demise, would he not have figured out that the triangular formations he had illustrated were junctional presynaptic regions, and jumped from the camp of the reticularist to the neuronist? Perhaps in such case he would have formulated the neuron doctrine before Cajal. Another militant in the reticularist camp was Joseph von Gerlach. Joseph von Gerlach was one of the first medical researchers who used microphotograph for scientific aims in basic tissue research. He considered his microphotograph to support the reticular theory. So, we have talked about three prominent reticularists. I will now say a few words about the players in the neuronist camp. In addition to Santiago Ramón y Cajal and his disciples, which included personalities of the stature of Lafora, who described myoclonic epilepsy with Lafora bodies, and many other of his pupils, we have Wilhelm His. William His made great contribution to the neuron doctrine. William His' main work was in the field of neurobiology. He observed the neural tube and correctly interpreted its function, and he also discovered the neural crest cells. Implicit in William his writing is the idea that there is contiguity but not continuity between nerve cells in the central nervous system. But his histological method did not permit him to demonstrate it. The last neurologist I will mention in this section is August Henry Forel. His contribution to the neuron doctrine was very significant. In this frame you can see two drawings. One of a normal facial motor nucleus of a guinea pig with well-formed neurons and axons, and another one after the facial nerve had been cut for a few days. Notice that the neurons have lost their angularity and axons are not present in most of the tissue. This observation, coupled with the lack of involvement of the neurons from the brain reaching the facial motor nucleus of the guinea pig, and other similar findings in other preparations, led him to the conclusion that there was a gap between the upper neuron to the facial motor nucleus and the lower motor neuron in the facial motor nucleus. He came to this conclusion because when a peripheral facial motor axon forming the facial nerve is injured, the neuron from which the axon originates dies, but not the one connecting to it. If no gap were present, Forel surmised, the injury to a distal neuron axon would not only involve the body of the injured neuron, but also the one above, connecting it. He went on to support the neuron doctrine with the following words. Ultimately, the branching always takes the shape of extensively ramified, interlocked trees, but anastomosis is found nowhere. I like to finish talking about Forel by telling you a little bit about his life. Forel, before becoming a great neuroscientist, was a great myrmecologist. I had to Google this word. It means a man that studies ants. In his early 20s, he wrote a 3-part book about the social life of Sweden ants. Here I have introduced this photograph of Forel holding one of his beloved ant collections. I have done so because I want to call your attention to his signature. Notice that the letters look irregular and do not flow smoothly from one to the other. Forel, at age of 52 years in 1912, had a stroke that left him with a right hemiparesis. The signature you see in this photograph was written years after the stroke with his left hand. It is freaky that in a painting of Forel 18 months before his stroke, Oskar Kojaska captures some features consistent with right hemiparesis that later afflicted Forel, especially the abnormal right hand position, as you can see here, held flexed at the wrist, and the flexion of his fingers. These features, noted by his family once the painting was finished, led Forel to decide not to buy the painting. But after the stroke, once the abnormal hand posture developed, Forel reconsidered buying it, but by that time the painting had already been sold. With this story, I will end our discussion of the neuron doctrine. And again, I will pose the animal question to you. To which of these animals the progress of neuroscience owes the most? Based on this section, I will go with C. The last landmark we will talk about is the ripples of the brain. We will approach this landmark using the lens of history, but introducing the characters in this segment at the same time. The four neuroscientists in this segment, which could very well label this saga, are Richard Caton, Adolf Beck, Vladimir Pravdich Neminsky, and Hans Berger. All of them lived through World War I. Adolf Beck, Vladimir Pravdich Neminsky, and Hans Berger lived through the turbulent times of World War II, and Vladimir Pravdich Neminsky, in addition, lived through the Russian Revolution and the Ukrainian-Russian War. As I was researching for this segment, I could not help but to think about a Chinese curse. May you live in interesting times at an interesting place, to which I added a Cuban blessing, and be unscathed by them. You will see why I had these thoughts as we go through the life of the neuroscientist in this section. Richard Caton was a world traveler scholar. His favorite destination was Egypt, and his favorite scholastic subject was Egyptian medicine. On the topic of Egyptian medicine, he spoke in multiple occasions. One of these occasions was at the Royal College of Physicians, where he was the keynote speaker for the Harveyan oration, named in honor of William Harvey, who I mentioned at the beginning of this talk as the British candidate for the discovery of blood circulation. In this oration, which took place in 1904, Richard Caton spoke about the prevention of valvular disease and of Imhotep, an ancient Egyptian doctor that was transformed from human to God because of his medical acumen. This lecture was later published in a widely read book of the same title. Richard Caton, in addition to lecturing in medicine and on history of medicine, had a very active social life. He became Lord Mayor of Liverpool in 1907. In his tombstone, you can see written who he was: a man greatly loved. As a neuroscientist, he accomplished much, all related to a single set of experiments, aspects of which he presented in 3 different occasions. The first of such occasion was prompted by a paper read by Dr. David Ferrier to the Royal Society in 1874 and illustrated by Ernest A. Waterlow. This paper was about the dog and macaque brain functional localization. The concept of cerebral functional localization propelled Richard Caton to present on the 4th of August of 1875 at the physiological section of the British Medical Association meeting in Edinburgh the findings of the experiment he had conducted in 1874. The presentation was summarized in a communication published in the British Medical Journal in August 28, 1875, as we previously mentioned, where he described its principal results. These results were: every animal brain studied generated electrical current. The injury potential found before by Matteucci in muscle and Dubois in nerves was also present in the brain. Brain currents of lower magnitude than the injury potential were detected with both electrodes on the brain surface, or if one electrode was in the brain surface and the other on the surface of the skull. The electric current of the gray matter appears to have a relation to its function. Brain activity produces local negative variations, and light influences brain currents. The second occasion was 2 years later, when he published a more precise explanation of his experimental methods and findings in 1877. This publication was in the British Medical Journal, and it was titled Interim Report on the Investigation of the Electrical Currents of the Brain. In it, he described the animals and the instruments used in his 1974 experiment. The animals used were rabbit, cats, and monkeys. The use of animals for experimenting brought upon him the disapproval and scorn of the members of the anti-vivisection movement, as reflected in this illustration from the time. As far as material, he used the electrodes built by Dubois Raymond and Thomson's reflecting galvanometer that projected the tracing of the brain electrical fluctuations into a scale as illustrated in this frame. The conclusion revealed in this article did not provide any new information regarding brain electrical currents per se, but they did reveal new findings about the relation of electrical currents and brain function, such as that the wake and sleep brain wave pattern were different, that anesthesia produced electrical current changes in the brain, and that brain currents fall to near zero after death. The third occasion he presented the result of his 1874 experiment was 10 years later in the 9th International Medical Congress in Washington, D.C., where he read a paper entitled Researches on Electrical Phenomena of Cerebral Gray Matter. It seems that in this lecture he did not put forward any new information but crystallized some of the previous observations, such as that cerebral electrical currents increase during the suspension of functional brain activity, and its corroborating finding that cerebral electrical current diminished when the cortical area examined is active. At this point, I'd like to ask you a question. Was Richard Caton the first experimentalist to record brain electrical activity? The answer, according to Lord Cohen of Birkenhead, is no. In Lord Cohen's article presented in March 4, 1959 and printed in the Proceeding of the Royal Society of Medicine, Lord Cohen stated that Richard Caton, at the time of his experiment, was aware of a paper which I had not been able to find despite spending a long time Googling it, written by Dubois Raymond, titled The Electrical Current of the Frog's Brain, in which Dubois describes the presence of injury potentials in frog's brain, and a second paper titled Experiment on the Physiologic Action of Light by James Dewar and J. G. McKendrick, which I was able to find in the collection of papers of Sir James Dewar put together posthumously by Lady Dewar, published in 1927. As you can see in this frame, I have introduced a segment of this publication, which was reproduced from the Proceedings of the Royal Society of Edinburgh in 1873, 1 year before Richard Caton's experiment, and 2 years before his first communication, where it states that the effect of light on the retina has been traced into the brain in the form of current in the optic lobe, referring to the occipital lobe. The next neuroscientist we will address is Adolf Beck. His life was a dream that turned into a nightmare. Of the 3 neuroscientists left in this section, he's one of the 2 whose life ended in suicide. Adolf Beck was born and worked in Poland. He was Jewish. And with the rise of the Nazi Party and the persecution of Jewish people, initially confined to Germany, but later expanded to Poland, Adolf Beck's life was turned upside down. Initially, Adolf Beck was able to evade the concentration camps with the help of his son, Henryk, and a Polish couple, Sofia and Krzysztof Wileński. But when he became ill and went to the hospital, he was captured. Just before being taken away by the Gestapo, his son, Henryk, gave him a capsule with cyanide, which Adolf Beck took before hearing the tango of death. His grave was never found. Adolf Beck's contributions to neuroscience were prominent. Using Dubois-Reymond electrodes and a more potent galvanometer than the one used by Richard Caton, he was able to capture the brain currents from electrodes on the surface of the brain of rabbits. He recorded from the occipital region an ongoing rhythmical electrical activity of fluctuating voltage that, after a flash of light, was interrupted by a high voltage wave called an evoked potential, and followed by flattening of the previously ongoing activity, which is called desynchronization. He published these findings in a distinguished physiology journal. After this publication, Adolf Beck was credited with having been the first to describe electroencephalographic activity and evidence of sensory modalities localization of the cerebral cortex, the first to record an evoked potential, and the first to describe the synchronization following stimulation. Shortly after this publication, a controversy arose regarding the ownership of these credits among 3 neuroscientists living in Central Europe: Adolf Beck in Krakow, Poland, Ernst Friedrich von Marksow in Vienna, and Vasily Yakovlevich Danielewski in Kharkov. Their squabble about priority can easily be followed in the Central for Physiology journal. Adolf Beck published his findings in the November 8, 1890 issue. In the December 6, 1890 issue, Ernst Friedrich von Marksow published an article which stated that he had done similar experiments, had obtained similar results, and has written a report which he kept in a sealed letter dated 1883, safe in the Imperial Academy of Science in Vienna. In addition, to prove his claim, Fleischer reproduced the letter in his publication. A third paper published claiming having recorded animal brain waves before was published on 11 April of 1891 by Professor Danielewski. In this paper, Danielewski called attention to his 1876 doctoral thesis titled Investigations in the Physiology of the Brain, where he described spontaneous and provoked currents present in dog's brain and provided a description of the fluctuating brain potentials and desynchronization. So how did this squabble end? It ended with another publication in Certain Blood for Physiology, this time by Richard Caton. In it, he reproduced his 1875 British Medical Journal communication and commented, "It is not my purpose to belittle the fame of the learned physiologist," referring to Beck, Fleisch, and Yakovlevich. "Still, I did make these discoveries myself, and as I mentioned above, have published them, so I think it will be admitted that I was already the earlier discoverer." The third neuroscience I will mention in this segment is Vladimir Pravdich-Neminsky. Vladimir Pravdich-Neminsky, while being medical student in Kiev, using an Einthoven stir galvanometer and non-invasive surface electrodes, recorded dog's brain electrical activity from the scalp surface, not with at least one electrode on the brain surface, as it had been done by neuroscientists before, but he published his findings on the 29th of November 1913 on the Central Blood for Physiology journal that all the other physiologists have been publishing their own articles. Here you can see two of the figures in this article. These are the first recording of electrical cerebral waves using non-invasive techniques ever published. Shortly after this publication, events took a hold of Vladimir Pravdich-Neminsky's life, which halted further experiments. First came World War I, then Lenin coming to power in Russia, then the Ukrainian-Soviet War, and then Stalin. All these events influenced Pravdich-Neminsky, but not as much as Stalin's reign. In 1929, he was accused and found guilty of lacking communist credentials and participating in counter-revolutionary activities. As a result, he was deported from Kiev to Arkhangelsk, up in the northern part of Russia. Months later, he escaped and went to Moscow, where he apparently lived as a fugitive for a long time. In 1949, Pravdich-Neminsky accusations were dropped, and he was allowed to work. He later became the head of the Laboratory of Cerebrography at the USSR Academy of Science. He held this post until his death. The fourth and the last neuroscientist I will talk to you about is Hans Berger. Hans Berger published in Archive of Psychiatry and Neurological Sciences in 1929, the same year Pravdich-Neminsky was sent to captivity close to the Arctic Circle, a paper titled About the Electroencephalogram in Men. In it, he published several tracing of human EEG recorded by using scalp electrodes. Here you can see reproduced three of these recordings. Hans Berger was the first to record the basic rhythms of human brains, the ripples of the brain. The impetus to start his experiment on electrical brain activity was triggered by a fall from a horse. Hans Berger, at the age of 19, enlisted in the German army. 1 year later, in a spring morning, Hans Berger was involved in a military exercise. During it, Hans Berger had a near-fatal accident. He fell from his horse and was about to be run down by a horse-drawn cannon, but the driver of the artillery battery halted the horse in time. That night, Berger received a telegram from his father inquiring about his well-being. This telegram was sent on the insistence of Berger's sister, who had been overwhelmed by a sense that something grave had befallen her brother. The timing of her sense of doom coincided with the time of Berger's near-missed fatal event. Berger later wrote about this incident, describing it as a case of spontaneous telepathy. He explained it by saying that at the time of mortal danger, as he contemplated certain death, he transmitted his thought, while his sister, who was particularly close to him but many miles away, acted as the receiver. It was this incident that led Hans Berger to postulate that the cerebral cortex had psychic energy along with heat and electrical energies. He initially considered building a machine to record psychic energy, but desisted. Had a brush at building a machine to record brain heat energy, but finally settled for looking for electrical energy. He started recording brain waves in 1924, first in patients with skull defects, then in individuals with intact skulls. In this figure, you can see one of Berger's patients being recorded. Notice, for this early experiment, he used Dubois electrodes. Later on, he started creating his own electrodes. The publication I mentioned before, his first on the subject of EEG in men, was not met with general approval. Neither did his second, third, or fourth publications on the same subject. But after the fourth publication, in 1933, things started to change. But it was not until the publication by E.D. Adrian and B.H.C. Matthews of a paper in Brain titled Berger Rhythm: Potential Changes from the Occipital Lobe in Man that Berger findings were acknowledged as a great discovery, and his 1929 publication was recognized as a landmark article in the history of neurophysiology. For just about the next 10 years, until 1938, Berger continued writing on the subject. All in all, he published 14 papers with the same title, On the Electroencephalogram of Man. In them, we find recordings of normal brain activity. Here you can see the recording of his own daughter, which he labeled the electrical rhythm according to their frequency as alpha in the 8 to 13 cycle per second, present at rest, beta above 13 cycle per second, recorded while his daughter was engaged in mathematical calculations, and a mix of beta and alpha wave while she was giving the result of the calculations. In addition, Hans Berger published recording of patients having generalized tonic-clonic seizures, petit mal, and dementia. Hans Berger had the misfortune of living during interesting times at an interesting place and not the luck to be unscathed by them. His behavior during the Nazi regime has been praised by some and then by others. A poem by Berger's maternal grandfather hung on the wall of his study that probably summarized who he was goes as follows. Each man faces an image of what he is meant to be. As long as he does not reach it, he cannot attain his full measure of peace. In 1941, Hans Berger committed suicide. So we have addressed the last neuroscience landmark, and I'd like to end this talk by reintroducing the animal question I had posted to you a few times during this talk. To which of the following animals the progress of neuroscience owes the most? Each of you can choose one. I choose the frog. Thank you very much for your attention.